Good evening and welcome back. Find a position that suits you. Let your shoulders drop a little and set your sleep timer if you use one, because nothing here needs to be followed closely. Tonight, the story is about blood and about a letter of the alphabet that ended up printed on millions of cards, in millions of wallets, in almost every country in the world. It begins in a laboratory in New Jersey in the summer before the war, with a doctor holding a glass tube up against a lamp, and it ends in an ordinary armchair in Australia in our own century with a man who did not like needles. There's nothing to solve along the way. Everything in this story has already happened, and it's already been written down, and we are simply going to walk through it slowly in the order it occurred. We are in Newark, New Jersey, in the summer of 1939, in a hospital laboratory where the work of the day is done in small glass tubes, and the answers arrive in the form of clouding. A woman had been admitted to the hospital and had given birth to a stillborn child. She was bleeding afterwards, as women sometimes did, and she needed blood, and the blood that was available and convenient was her husband's. By the standards of the time, this was a careful decision, not a careless one. Blood groups had been known for more than 30 years. The letters A and B and O were understood, and the husband and the wife were both Group O, which was as good a match as anyone in 1939 knew how to make. The transfusion was given, and the woman became ill in a way that nobody in the room could account for, with pain and with fever and with urine that had darkened, which was the old and unmistakable sign that red cells were being broken apart somewhere inside her. The physician caring for her was Rufus Stetson, and the man who was given her blood to examine was Philip Levine, who worked in the transfusion laboratory in Newark, and who had spent years already looking at the small quarrels that human blood can have with itself. What Levine did next was ordinary laboratory routine, which is exactly why it worked. He took the woman's serum, the pale straw-colored fluid that remains when blood is allowed to separate, and he mixed it, drop by drop, with red cells from other people. Every tube was the same size, every tube held the same small volume, and every tube was read the same way. Held up, tilted, turned slowly against the light, and judged. The tube either stayed clear or it clouded. Her serum was mixed with her husband's cells, and the tube clouded. The cells had clumped together, gathered into small visible grains, and settled. That, on its own, was strange but not unheard of. What made the afternoon unusual was what happened when Levine went further and tested her serum against the blood of a long series of unrelated donors. All of them matched to her by the letters that were supposed to matter. Roughly 8 out of every 10 of those tubes clouded as well. Something in the woman's blood was reacting against something carried by the great majority of the people around her, a thing present in most human beings and absent in her, which had no name, no letter, and no place in any chart then in use. The tubes themselves were made of soda lime glass, washed and reused, and stored upside down in wooden racks that were drilled with rows of holes and left unvarnished so they could be scrubbed. A serological laboratory of that period ran on saline solution, kept in large, stoppered flasks, on hand-cranked or small electric centrifuges, bolted to the bench so they would not walk, and on a single, strong lamp used for reading results. Records were kept in ledgers, in ink, 1 line per patient, and the results were written as a plus, a minus, or a small drawn cross. The two men wrote the case up, and it appeared in July of 1939 in the Journal of the American Medical Association under a title so mild that it is easy to read past it, an unusual case of intragroup agglutination. The paper is barely 2 pages long. It describes the patient, the transfusion, the reaction in the tubes, and it proposes an explanation that was, in the summer of 1939, a considerable step, that the woman had been immunized by her own pregnancy. That is, the child she had carried had possessed something in its blood inherited from its father, something the mother's body did not itself possess, and her body had learned to recognize it and to make an answer to it, an antibody which was still circulating in her months later, and which had then met the same thing again in her husband's transfused cells. The paper did not name the factor. It did not have to. It simply recorded that the thing existed, that it was common, and that it could be inherited from a father through a child and taught to a mother. And then it went quietly into the literature to wait. We are still in that period in the late 1930s and the early 1940s in rooms of this kind, and it is worth staying at the bench a while because the work is slow and the rhythm of it is the rhythm of the whole story. A serology laboratory before the war was a quiet place, and it was quiet by necessity because everything that happened in it was small. The unit of work was the drop. A technician drew blood into a tube, let it stand until the red part sank and the clear part rose, and separated the two with a fine pipette held between 2 fingers. The top of it closed with the ball of the thumb. Red cells were then washed. That meant adding saline, spinning the tube, pouring off the fluid, and adding saline again 3 times over, until what remained at the bottom was a small button of cells and nothing else. The washing took longer than the test. Nothing in the process could be hurried because the reactions were read by eye, and an eye that is hurried sees what it expects. A weak reaction and no reaction look very much alike, and the difference between them was, in some rooms, the difference between a safe transfusion and an unsafe one. The reagents came from animals to find out whether human blood carried a particular marker. A laboratory needed a serum that would react to that marker, and the reliable way to obtain one was to inject an animal, a rabbit, a guinea pig, with the cells in question, wait for the animal to build its answer, and then take that answer, purify it, and keep it in small labeled bottles under refrigeration. Those bottles were treated with a care that is hard to overstate. A good anti-serum was scarce. It could not be bought at short notice, and if a laboratory lost one, it could take months to make another. Some were named after the animal that produced them and were spoken of almost as colleagues. Blood itself, in those years, was kept in glass bottles rather than plastic bags, sealed with rubber and stored in domestic-style refrigerators that were checked by thermometer and noted by hand. The anticoagulant was a citrate solution, first used at scale during the previous war, which kept blood liquid outside the body for a matter of weeks rather than hours. A bottle had a paper label, a date, a group written large, and a piece of string or tape to mark the cross-match, and it was carried to the ward upright in 2 hands, like something hot. There was also, in those rooms, a very particular relationship with the word unexplained. Transfusion in the 1930s worked most of the time, and when it did not work, The failure was usually put down to one of a small number of familiar causes: a clerical error, a mismatched group, a contaminated bottle, an over-rapid infusion. But a residue remained. In every large hospital, there were reactions that had none of those explanations, that occurred between people who were properly matched by every letter then known, and that were written into the ledger with a note and left there. The tube either stayed clear or it clouded, and in a certain small percentage of cases, it clouded when, according to everything on paper, it should not have. That residue is what Levine's 2-page paper walked into. It did not remove the mystery so much as give it a shape, and it suggested for the first time in print that the shape was inherited and that pregnancy could be the teacher. We are a few months further on now, in 1940 in New York, in a different laboratory, in a different line of work that was about to cross the first one almost by accident. Carl Landsteiner was by then an old man in scientific terms and a famous one. 40 years earlier, in Vienna, he had been the first to describe the ordinary blood groups, the letters that let transfusion become a routine procedure instead of a gamble, and he had been given the Nobel Prize for it a decade before this story begins. He was working in New York with a younger colleague, Alexander Wiener, and their question that year was not about human beings at all. It was a comparative question of the kind that occupied a great deal of laboratory time in that period. How far do the blood markers of other primates resemble our own? To find out, they took red cells from a Rhesus macaque, a small brown monkey of southern Asia, then the standard laboratory primate kept in numbers in animal houses across the United States, and injected them into rabbits and guinea pigs. The animals did what animals do. They produced antibodies against the monkey cells, and after a suitable interval, the researchers drew off the serum containing those antibodies and had in their hands a reagent that would react against something carried on Rhesus red cells. Then they did the obvious next thing, which was to see what that reagent made of human blood. It clouded roughly 85 tubes out of every 100. The great majority of the New Yorkers whose blood they tested carried something that this monkey-derived serum recognized, and a minority did not, and the split fell in almost exactly the same proportion as the split Levine had found the year before in a woman's serum in Newark. The two findings were laid alongside each other, as such things are, and the conclusion drawn was that they were the same discovery, arriving from two directions. The factor was named for the monkey that had produced the reagent, the Rhesus factor, shortened almost at once, in speech and in writing, to the two letters R and H. Here the story does something that stories in laboratories do more often than is usually admitted. The conclusion turned out to be not quite right, and the name outlived the mistake. Within a few years, it became clear, through careful comparison of the reactions, that the antibody the rabbits had made against monkey cells and the antibody the woman in Newark had made against her own child's cells were not aimed at precisely the same target. They were aimed at two different structures that happened to sit close together on the surface of the red cell and to travel together in most people. The animal-derived one was quietly renamed decades later after the two men who had produced it. The human one kept the monkey's name. So the letters that are printed on donor cards and and hospital wristbands all over the world today, and that appear after the letters A, B, and O as a small plus or a small minus, are a memorial to a comparative experiment about primates that reached a conclusion which was afterwards corrected. The Rhesus macaque itself, meanwhile, went on to a laboratory career of its own, and its name is attached to more of 20th century medicine than almost any other animals. Colonies were maintained in the eastern United States and in India. Animals were shipped by sea and later by air in wooden crates with slatted sides, and the demand from research institutions was large enough that the trade became, for a period, a matter of diplomatic correspondence between governments. The monkey is gray-brown, weighs about as much as a large domestic cat, and in the wild lives comfortably alongside people in towns. By the end of 1940, then, two things were known that had not been known 2 summers earlier. The first was that most human beings carry a particular marker on their red cells, and a minority in the populations being tested at the time, roughly 1 person in 6 or 7, do not. The second was that a woman who does not carry it can meet it through a pregnancy and can learn to make an answer to it and can keep that answer in her blood afterwards. What had not yet been said out loud in print was what those 2 facts meant together for the child. We are stepping backwards for a short while, out of the laboratory and into a much older part of the record, because the illness in this story is far older than the letters that explain it. In 1609, in France, a midwife named Louise Bourgeois wrote an account of a birth she had attended, and the account was published because she was a woman of standing whose books on midwifery were read across Europe. She described twins. One of them was born badly swollen, with fluid gathered under the skin, and did not live. The other was born looking well enough, and then over the days that followed, turned a deep yellow and grew slack, and did not live either. She had no explanation for it, and she offered none. She wrote down what she had seen in order, the way the useful parts of the old medical record tend to be written, and she moved on to the next case. That description sat in the literature for more than 300 years without a mechanism attached to it, and it was not alone. Similar accounts accumulated slowly in Latin and in French and in German and in English of infants born swollen, or of infants who yellowed after birth and did not recover, sometimes one after another in the same family, sometimes with the second child worse than the first and the third worse than the second. That last pattern, the worsening, was noticed early and remarked on often. It was the most consistent thing about the illness and the least explicable. A first pregnancy would go well, a second would go less well, a third might not go at all. Families knew this before doctors could account for it, and in some regions it acquired local names that had nothing to do with blood. In 1932, in Boston, 3 physicians, among them Louis Diamond and Kenneth Blackfan, pulled several of these separate presentations together and argued that they were one condition seen at different depths: the swelling, the yellowing, the severe anemia of the newborn, and the appearance in the infant's blood of immature red cells that ought never to have left the marrow. Those immature cells gave the illness the name it carried for the next generation: erythroblastosis fetalis. It is a heavy phrase, and it means, more or less, an excess of young red cells in the unborn. So, by the middle of the 1930s, the illness had been given a single outline and a single name. What it did not have was a cause. Then came the 2-page paper from Newark and the monkey-derived serum from New York. And in 1941, Philip Levine, the same man, still working with the same kind of glass tube, published the connection. The mother lacked the factor. The child, having inherited it from the father, carried it. Cells from the child crossed into the mother's circulation, most reliably at birth, and her body learned them and made its answer. In a later pregnancy, that answer, which is small enough to cross the placenta in the other direction, reached a child who carried the factor and broke down that child's red cells before it was born. That is the whole of the mechanism, and it explains, without needing anything else, the one feature families had always noticed, why the first child was usually well and the later children were not. The factor itself, understood much later at the level of the molecule, is a protein that sits threaded through the wall of the red cell, passing back and forth across that membrane a dozen times, anchored in place by a second protein that keeps it company. People described as negative are, in the great majority of cases, simply people in whom the stretch of chromosome carrying the instructions for that protein is absent altogether, not altered, not damaged, but gone on both of the chromosomes inherited from both parents. The cell is built as usual, and the protein is left out. There followed through the 1940s an argument about names. 2 systems of notation were proposed for the growing family of related factors, one by Alexander Wiener in New York and one in Britain, worked out from the reaction tables by the statistician Ronald Fisher and taken forward by Robert Race. They divided the same reality in different ways, and they wrote it with different symbols, and each camp believed the other's notation obscured what was actually happening on the cell. The argument went on for years, in journals and at meetings, with a persistence that anyone who has worked in a specialized field will recognize. Both systems remained in use. In much of the world, the British lettering won the daily habit, and the principal factor, the one the woman in Newark had met, the one that matters most in pregnancy, came to be written simply as a capital D, which is why, in the end, the plus and the minus on a donor card refer to one letter of the alphabet, standing for one protein that most people have and some people do not. We are in the 1940s and 50s now, on the wards rather than at the bench, in the years when the cause was known and the remedy was not. Knowing the mechanism changed what could be predicted long before it changed what could be prevented. And for about 25 years, the practical work consisted of watching, of counting, and of replacing. A woman known to lack the factor was tested during pregnancy to see whether her blood had begun to make the answer, and the level of that answer was followed from month to month. Where it stayed low, the pregnancy was managed as any other. Where it climbed, the child was assumed to be affected, and the plans changed. The one real treatment of that era was replacement, and it was carried out after birth. A newborn's blood was withdrawn a little at a time and replaced a little at a time with donor blood chosen to lack the factor, so that the mother's antibodies, still circulating in the infant, would find nothing left to act upon. Technique is called exchange transfusion. It was developed in the mid-1940s, refined over the following decade, and it was by the standards of the time a demanding procedure. Slow, performed with syringes and stopcocks, requiring the infant to be kept warm and still, and requiring the person performing it to keep count aloud. It worked often. Something in the order of 50,000 infants in the United States received it in the decades before prevention existed, and a great many of them lived because of it. But it was a repair carried out after the event on a child who had already been affected, and it could do nothing at all for the pregnancies where the harm had been done too early, before the child could be born and reached. The equipment for an exchange in that period was laid out on a covered trolley in a fixed order, a series of glass syringes of graded size, metal three-way taps, rubber tubing cut to length, a warming water bath, cotton swabs, and a ruled paper chart on a clipboard for recording each volume in and each volume out. The infant lay on a heated table under a cloth. The room was kept warmer than the corridor, which is why nurses who worked in those rooms remembered them decades afterwards, chiefly as being too hot. Two further advances belong to those years, and both came from the same instinct. If the harm happens before birth, then before birth is where the work has to go. The first was a way of measuring. In New Zealand in 1961, an obstetrician working in Auckland found that the fluid surrounding an unborn child carries a faint yellow pigment when the child's red cells are being broken down, and that the depth of that color can be measured by passing light through a sample and reading how much is absorbed. That measurement, plotted against the week of pregnancy on a simple chart divided into 3 bands, told the physician for the first time not merely that a child was affected, but roughly how badly. It replaced a guess with a line on paper, and it was adopted quickly in Britain and North America and beyond. The second was a way of intervening, and it belongs to the same man working in the same hospital 2 years later, but that part of the story sits in the module ahead, and we will come to it in its proper place. What matters here is the shape of those 2 decades, because it is the least remembered part of the whole account. The cause was known. The mechanism was written down in ordinary language in the journals. The families understood the pattern, often better than they wished to, and for roughly a quarter of a century, the only answers available were to measure carefully, to deliver early when the measurements demanded it, and to replace the blood afterwards. The tube either stayed clear or it clouded, and by the 1950s, the clouding could be seen coming months in advance without changing very much about what would follow. We are in Liverpool now, at the end of the 1950s, in a city of docks and rain, and the story begins in a garden rather than a hospital. Cyril Clarke was a general physician, and he was also, seriously and at length, a breeder of butterflies. The 2 occupations were not kept apart, and in the end, they explain each other. His particular interest was mimicry, the way certain harmless swallowtail butterflies come to resemble other species that birds have learned to leave alone, and the way those resemblances are inherited in families of butterflies as neat, trackable packages. To study this, he needed to breed them in quantity through generation after generation, which meant a heated greenhouse, hand-pollinated food plants, and a great deal of patient pairing of one insect with another. His wife, Clarke, ran much of that work and was named on the papers that came out of it. It is worth pausing on what that hobby actually is, because it is not really a hobby. A person who breeds butterflies for inheritance keeps ledgers. He records which individual was crossed with which, what emerged, in what proportion, and how the traits traveled, and he learns by repetition to think in terms of what is carried, what is expressed, and what is hidden and passed and unseen. That is the same habit of mind that the blood factors require, and Clarke, in his 40s, turned it toward them. The Liverpool group around him included a young physician named Ronald, who had qualified locally in the mid-50s and who was working on the blood groups in relation to disease. The question they were circling was simple to state and hard to answer. The mother learns the factor from the child's cells, so is there any way to stop the lesson from being learned? Two pieces of knowledge lay ready to be joined, and both had been published elsewhere. The first was a staining method described in Germany in the late 1950s, which made it possible to take a drop of a mother's blood, treat it, and see under the microscope how many of the red cells in it had come from her child. That answered a question nobody had been able to answer before. Fetal cells cross into the mother in small numbers, and most of all at delivery. The second came from immunology, and it was already familiar in other contexts. If a person is given a ready-made antibody against a particular target at the same moment as the target itself arrives, the person's own immune system, for reasons that were then only partly understood, tends not to bother learning it. The answer has already been supplied, and the lesson is not taken. Put those together, and the proposal writes itself. Give the mother, at the moment the child's cells arrive, a dose of the very antibody she must never be allowed to make. The borrowed antibody clears the child's cells from her circulation before her own system can register them, and then the borrowed antibody itself fades away over weeks, leaving nothing behind. She remains immunologically as she was. The next pregnancy begins from a clean page. The antibody needed for this could only come from human beings who already had it, which meant at first from women who had been sensitized in exactly the way the treatment was meant to prevent, and who agreed to give plasma afterwards. Later, it came chiefly from volunteer donors, most of them men lacking the factor, who were deliberately given small quantities of red cells carrying it, and who then donated plasma at regular intervals for years. The plasma was pooled, the useful fraction separated out by cold alcohol precipitation, and the result issued in small glass ampoules, each holding rather less than a teaspoonful. Finn put the idea forward publicly in 1960 in a short note and at a meeting, and the Liverpool team began to test it. The first experiments were done on male volunteers because the design required people who could be given the child's cells in a controlled way and then watched. Cells carrying the factor were given to men who lacked it. Some of the men were also given the borrowed antibody, and some were not, and the blood of all of them was followed for months to see who had learned and who had not. The results were published in the British Medical Journal in 1961 under the plain title of Experimental Studies on the Prevention of Rh hemolytic disease, with 7 names on the paper. They were encouraging rather than conclusive, which is the ordinary state of a first result. But the principle held. The men who had received the borrowed antibody were slower to make their own, and in several cases did not make it at all, and at almost exactly the same time, without any coordination, and for the same reasons. The same idea was being tested 3,000 miles away. Chapter 7, New York and the Small Glass Ampoule. We are in New York in the early 1960s now, in a large teaching hospital in the north of Manhattan, where 3 men were arriving at the same proposal from 3 different directions. John Gorman ran the blood bank there. He was Australian by birth and training, and had come to the hospital at the end of the 1950s, and it was he who made the connection between the immunological principle and the obstetric problem. Vincent Freda was an obstetrician in the same institution, which meant he was the one who saw the pregnancies themselves year after year, and who knew precisely what the existing management could and could not do. The third was William Pollock, who was not a hospital man at all. He was a protein chemist working for a pharmaceutical company in New Jersey, and his contribution was the least visible and the most necessary. He knew how to take pooled human plasma and pull out of it, cleanly and repeatably, the single fraction that carried the antibody. Without that, the idea remained a proposal. An antibody that cannot be purified in quantity to a consistent strength in a form that can be injected safely is a laboratory curiosity. The first human tests in New York were run in the early 60s in male volunteers held at a state prison on the Hudson River, north of the city. The men lacked the factor. Some were given the antibody and some were not. All were then given cells carrying the factor and followed with monthly samples. It is worth saying plainly, because the record says it plainly, that this kind of prison research was ordinary practice in that decade and would not be permitted now, and that the standards which govern such consent today were written in part because of the studies of that period. The results matched Liverpool's. The men who had received the borrowed antibody did not learn the factor. A preliminary report was published in 1964. In January of that same year, the first woman at real risk received the injection, a member of Gorman's own family, his sister-in-law, which tells its own quiet story about how confident the 3 men were by then. What followed was neither quick nor sudden. It was a large, slow, coordinated trial. Dozens of clinical centers in the United States and Britain and Germany and Sweden and Italy and Australia, each enrolling women who lacked the factor and had just given birth to a child who carried it, each giving the injection within days of delivery, each following the mothers afterwards to see whether the answer had been learned. The trial confirmed the principle at scale, and in 1968, the preparation was approved for use in Britain and in the United States. The dose approved was 300 micrograms of purified antibody, given by injection into the muscle within 72 hours of the birth. 300 micrograms is a quantity difficult to picture. It is roughly 1/3 of 1,000th of a gram, less than the weight of a single grain of fine table salt. Suspended in a volume of fluid, smaller than a sugar cube, it is given in the upper arm or the outer thigh, takes a few seconds, and is unremarkable in every way except one, which is that it is one of the least expensive interventions in the whole of modern medicine. There is a small, exact detail recorded from the first day of its availability in the United States in June of 1968, and it is the kind of detail that survives because someone wrote it down at the time. a woman named Marianne Cummins, then 29 and expecting her third child, had read in a newspaper that the new preparation would be released on the 1st of June. Her own due date was the 27th of May. The arithmetic mattered because the injection only works if it is given within about 3 days of the birth. She was, by a few days, on the wrong side of the calendar. She delivered late as it happened and received the injection within hours of the preparation becoming available. The first person to be given it outside a trial anywhere in the world. 50 years afterwards, she was still there to tell the story herself. At a gathering held to mark the anniversary, sitting alongside the physician who had given it. Within about a year, the injection had been given to more than 500,000 women. Within a few years, in the countries that adopted it, an illness that had been ending the lives of something like 10,000 infants a year in the United States alone had become an uncommon thing that most young obstetricians would see only in textbooks. Chapter 8, Auckland and the work done before birth. We are moving to the other side of the world for this chapter, to Auckland in New Zealand, and stepping back a few years, because while Liverpool and New York were working on prevention, a third line of work was running in parallel, and it concerned the children who could not be prevented because their mothers had already learned the factor years before. The obstetrician in question, William Liley, worked at the National Women's Hospital in Auckland, and it was he who had devised the yellow pigment measurement described earlier, the chart with 3 bands that told a physician how badly an unborn child was affected. Having a measurement raised an obvious and uncomfortable question. If the chart showed a child in serious difficulty at 26 weeks, and the child could not safely be delivered until much later, the measurement changed nothing at all. It only told you sooner. The idea he arrived at in 1963 was that a transfusion could be given to the child where the child was. The route he chose was the abdominal cavity rather than a vessel, because a vessel in an unborn child of that size could not be reached with the equipment of 1963, and because he had learned from a visiting doctor describing practice with small children elsewhere that red cells placed in the abdominal cavity are gradually absorbed into the circulation over the following days. The technique in outline was to pass a needle through the mother's abdominal wall and the wall of the womb, confirm the position by radiograph after injecting a contrast medium, and then introduce donor red cells lacking the factor slowly through fine tubing. The first 3 attempts did not succeed. The 4th did, and the report appeared in the British Medical Journal in November of 1963. The response was immediate and international, and it is a useful illustration of how fast a printed technique could travel even then. In Winnipeg, in central Canada, a physician read the November issue and, with a colleague, attempted the procedure in January of 1964. That first attempt did not succeed. The next one, the following month, did. A woman in her 4th pregnancy who had lost 2 children previously and whose child received 4 transfusions in the womb across the second half of the pregnancy and was born alive. The early success rate was modest, somewhere around 4 cases in 10, and everyone involved said so in print. It improved, decade by decade, as the equipment improved. The procedure, as it exists today, is done under continuous ultrasound guidance through a needle finer than the ones used then. The target is no longer the abdominal cavity, but the vein of the umbilical cord where it enters the placenta, a vessel a few millimeters across, and a moving subject entered from outside the mother's body. The blood used is washed, irradiated, closely matched, and warmed. In the hospitals that do this work, the same small team performs the procedure for the whole region several dozen times a year, and the room is kept quiet by convention. So, by the end of the 1960s, 3 separate things existed that had not existed at the start of the decade. There was a way to prevent the mother from ever learning the factor, given as an injection after birth and before long, during pregnancy as well. There was a way to measure how badly an already affected child was doing, week by week, and there was a way to treat that child before it was born. The illness did not disappear. It became, in the countries that could afford these 3 things, an uncommon event with a well-worn set of responses, which is the quietest kind of ending a medical story can have, and the most complete. We are leaving the hospitals now for the rest of the account and turning to something slower, the distribution of this trait across the world, which is uneven in a way that has interested population geneticists for 80 years. The first thing to establish is the ordinary background. Across the world as a whole, the great majority of people carry the factor, and something in the region of 6 in every 100 do not. That figure, however, hides almost everything interesting because it is an average of populations that differ from one another very considerably. In Europe, the proportion lacking the factor runs at roughly 15 in every 100, and it has been measured at that level with local variation from Ireland to the Baltic for as long as such surveys have been done. In much of East Asia, it is very different. Large population studies in China have returned figures well under 1 in 100, and Japanese figures are of the same order. Among indigenous populations of the Americas and of Oceania, the proportion is similarly low. Sub-Saharan African populations vary, generally at a few per 100, while parts of North Africa run considerably higher. And then there is one region where the figure departs from everything around it. Along the western end of the Pyrenees, on both sides of the mountains in the country of the Basques, the proportion of people lacking the factor has been measured repeatedly at around a third. That is roughly double the European figure, and something like 50 times the East Asian one in a population that occupies an area a person could drive across in a morning. The observation is old. It was made in the 1940s, when blood group surveying was a fashionable and inexpensive way of comparing populations. And it was immediately noticed that the Basques were also unusual in other respects, chiefly in speaking a language that is related to no other language on earth. Those 2 facts were joined together very early and have been joined together in popular writing ever since, and it is worth being careful about the joining. The language is genuinely an isolate. The blood figure is genuinely a high point, but a high frequency of a common European variant is not the same kind of statement as an unrelated language, and the 2 do not require the same explanation. There is also a measurement problem that quietly inflates the figures quoted in general writing, and it is worth stating once. A survey can report the proportion of people who show the trait, or it can report the proportion of the underlying instructions carrying the deletion. And these are different numbers, because a person can carry one copy without showing anything at all. Modern sequencing work in Basque samples has reported the second kind of figure at well above 40 in every 100. Older serological work reported the first kind at rather less. Summaries that give a single wide range are usually two different measurements pressed together. The explanations offered for the peak have changed as the tools have changed. The oldest was isolation. The Western Pyrenees are mountainous. The population is long settled, and a group that mixes less with its neighbors will drift away from them in the frequencies of whatever it happens to carry, a process that requires no advantage at all, only time and a degree of separation. Later work added the observation that the region sat within one of the areas of southwestern Europe, where human populations persisted through the coldest phase of the last ice age, which gives the isolation a great deal more time to operate in. More recent genomic work has complicated the older picture in an interesting direction. It suggests that the Basques are not a population that stood outside European history untouched, but one that received the same major waves of settlement as its neighbors, and then mixed comparatively little afterwards, which is a different thing and a more ordinary one. There is also a genuine puzzle in the arithmetic, and it is the most elegant part of this chapter. A variant that causes difficulty in pregnancy, as this one does, when a mother lacking it carries a child who has it, ought to be pushed downwards by that difficulty and to become rare. Instead, it sits at high frequency across a whole continent. The mathematics of this has been worked through, and the resolution is quietly satisfying. The disadvantage is strongest when the variant is rare, and it weakens as the variant becomes common, because a common variant means both parents are more often alike, and the mismatch arises less often. At around half, the pressure is close to nothing at all, and drift, the ordinary random wandering of frequencies in a finite population, is then quite sufficient to hold it wherever it has arrived. We are back among glass and refrigeration for this chapter in 1961 in Australia for the strangest single observation in the whole of this record. And as is usually the case, it arrived during routine work. A sample of blood was being typed, and it failed to react with anything, not merely with the antibody against the principal factor, the one that the letters on a donor card refer to. It failed to react with the antibodies against every other member of the same family of markers as well, the whole set, of which the principal one is only the best known. The sample belonged to an Aboriginal Australian woman, and the finding was written up in the Lancet by a small group of researchers under a title as flat as Levine's had been 22 years earlier. A sample of blood with no detectable antigens of that system. The reason this was startling to the people who read it is that the proteins in question are not decorative. They sit threaded through the wall of the red cell, and they contribute to its structure. And the general assumption had been that a cell built without any of them would not work, and that a person built entirely of such cells would not be viable. The woman was, by every account, in good health. Since that first case, fewer than 50 people worldwide have been identified with the same absence across every continent and every population group, with the estimated frequency put at something like 1 in 6 million. The nickname the phenotype acquired in transfusion circles is golden blood, and the name is practical rather than poetic. Red cells carrying none of these markers cannot provoke a reaction against any of them, which makes such a unit uniquely useful to a small number of patients elsewhere in the world who have unusual variants of their own and for whom ordinary blood is unsafe. The reverse, however, is the hard part. A person with this blood can receive only the same from one of the few dozen known donors, and units of it are held frozen in a handful of rare blood banks and shipped internationally when a case arises. It is not in itself an advantage. Absence carries a mild anemia and a set of subtle changes in the shape and fragility of the red cell, described in the literature as a syndrome and generally living quietly in the background of an ordinary life. The frozen rare blood banks are an unglamorous piece of international infrastructure and have existed since the 1960s. Units are prepared with a cryoprotectant, held at deep freeze temperatures for years, and washed free of that protectant before use, which takes about an hour of laboratory work per unit. There is a register in which the rarest phenotypes are recorded and a small number of institutions that hold the reference sera against which unknown samples are checked. It is a network run largely by correspondence between a few hundred people, most of whom have never met, and it moves perhaps a handful of units of any given rarity in a year. There is one more absence worth putting alongside it, because the two are often confused. The people this whole episode has been about, those described as negative, are missing one thing: the instructions for a single protein on both inherited copies. Everything else about the red cell is entirely as usual. The people found since 1961 are missing the whole family. The difference between the two is roughly the difference between a house without a chimney and a house without any roof at all, and only the second is genuinely rare. The tube either stayed clear or it clouded, and in the very rarest samples of all, it stayed clear against everything the laboratory had on the shelf, which is how they were noticed in the first place. We are in the present day for this chapter, and in the most ordinary setting the story has yet visited, which is a clinic waiting room on a weekday morning. The path by which a letter and a sign arrived on a card in a wallet is worth following slowly, because it took most of a century and almost nobody noticed it happening. For the first decades after the war, knowing a person's group was something done when it was needed, which meant before an operation or before a transfusion, and the result lived in the hospital's own records rather than with the patient. Blood banking changed that as collection became organized on a national scale and as blood moved between cities rather than between beds in the same building. The labels had to become standard, the records had to become portable, and the person had to be identified reliably at the moment the bottle was hung. That is where the wristband comes from, and the two independent checks at the bedside, and the small printed card that donors were given to carry. The card was originally a convenience for the service rather than for the donor. It let a regular donor be recognized quickly, and it let the service call on particular groups when stocks of those groups ran low, which they did seasonally and predictably in the weeks after Christmas and in high summer. The second route by which the litters reached ordinary life is the one that concerns this story more directly, and it runs through pregnancy. By the 1970s, in the countries that had adopted the injection, testing every pregnant woman for the factor had become part of the first appointment, because the injection can only be offered to a woman who is known to lack it, and she can only be known to lack it if she is tested. So the blood taken at that first visit is checked for the ordinary letters, checked for the factor, and screened for antibodies that might already be present. 3 questions from one small tube, asked of every pregnancy in every year, in every clinic that can afford to ask them. That is how a laboratory finding from 1939 became a routine appointment, and how a great many people first learn a fact about themselves that they will then repeat when asked for the rest of their lives. The testing itself no longer involves the glass tubes of the earlier chapters. Most laboratories now use small plastic cards holding a row of narrow columns filled with gel, into which the cells and the reagent are placed together and then spun. Cells that have clumped stay high in the column, and cells that have not traveled to the bottom. So the result is read as a position rather than as a cloudiness. A single card handles several questions at once. The reading is done by machine, and the cards are stacked in trays of 50. The principle underneath is precisely the one from 1939. There is a small piece of memorial arithmetic hidden in the calendar too, and it belongs here rather than anywhere else. The day set aside internationally for blood donors is the 14th of June, and the date was chosen because it is the birthday of Karl Landsteiner, the same man who in his 70s in New York was injecting rabbits with monkey cells, and finding that 85 tubes and 100 clouded. He is commemorated for the earlier discovery, the letters A and B and O, which is reasonable, since that is the one that made transfusion possible at all. The later work, the one that carries a monkey's name and a correction, sits underneath the plus and the minus that follow those letters, and is not commemorated by anything except its own continued use, which is, in the end, the ordinary fate of a solved problem. The solution becomes a routine, the routine becomes an appointment, the appointment becomes a line on a card, and the card goes into a wallet and stays there for 40 years, being produced occasionally at a desk and put away again. We are coming to the last part of the story, and it takes place mostly in ordinary rooms over 60 years in Australia. A boy of 14 had a lung removed in the early 1950s and needed a great deal of donated blood to come through it, and spent a long time afterwards in hospital thinking about where that blood had come from. His name was James Harrison. He was born in a country town in New South Wales in the winter of 1936, worked for the state railways as a clerk, and disliked needles for his entire life, which he mentioned freely, and which never appeared to affect his behavior in the slightest. At 18, in 1954, he began donating blood, as he had decided in hospital that he would. Some years later, it emerged that his plasma contained an unusually strong and persistent quantity of exactly the antibody the injection is made from, probably, it is thought, acquired from the transfusions that had saved him as a boy. From that point on, he donated plasma rather than whole blood every 2 weeks for the next several decades. The number recorded at the end is 1,173 donations from 1954 until his retirement from the panel in 2018 at the age of 81, which was not his decision, but the age limits. He died in his sleep in a nursing home on the New South Wales coast in February of 2025 at the age of 88. The National Blood Service credits his plasma with contributing to the protection of something over 2 million children, which is the kind of figure that is impossible to feel and worth stating anyway. He was consistently reported as saying that his donations were no more important than anyone else's. It is worth being exact about what he actually did, because it was not heroic in shape. He drove to a donation center, sat in a chair for the better part of an hour every second week for 64 years, read something, drank the tea afterwards, and went home. All but a handful of those donations were taken from the same arm. Australia maintains a panel of around 200 such donors, whose plasma supplies the injections given to some 45,000 mothers a year in that country alone. Comparable panels exist elsewhere, and in recent years, laboratory-made versions of the antibody have moved into trials, which may in time make the panels unnecessary. The routine that surrounds a plasma donation has been essentially the same for decades. A short questionnaire, a drop of blood taken from a fingertip to check the iron, a chair that reclines, a warm blanket that is offered whether or not it is wanted, and a machine that separates the plasma and returns the rest, which takes rather longer than giving whole blood and leaves the donor cooler than they expect. Afterwards, there's tea and a biscuit and a required 10 minutes of sitting still. The tea is a rule rather than a courtesy. And what of the trait itself? The thing on the card, the small minus sign after the letter. It is an absence rather than an addition, a stretch of chromosome carrying the instructions for one protein is missing on both copies, and the red cell is built without it. Nothing in the documented record attaches any ability, any advantage, or any inheritance from elsewhere to that absence. It is one of the many ordinary variations that human populations carry, distributed unevenly for ordinary reasons of isolation and time. What is genuinely unusual about it is not the trait, but its history. Of all the small variations in the human body, this is the one that provoked 100 years of laboratory work, gave its name to a monkey experiment that turned out to be pointing at something slightly different, and ended in a syringe holding less than the weight of a grain of salt. The people who now receive that injection at 28 weeks and again after birth, in clinics in every country that can supply it, are for the most part not told any of this. They are told that it is routine, which it is, and that it takes a moment, which it does. There is a newer development that belongs at the very end, quietly. In several countries, it is now possible to test a fragment of the child's own material, floating freely in a sample of the mother's blood, and to establish before birth whether the child carries the factor at all. Where the answer is no, no injection is needed and none is given, which means that the last chapter of this story is about not doing something, about a small number of women each year who are spared a routine that was itself designed to spare them. The woman in Newark is not named in the paper. She is described in the third person in a few lines in the summer of 1939. Everything that followed, the monkey serum, the letters, the arguments over notation, the greenhouse in Liverpool, the Ampoule, the needle in Auckland, the man in the chair every second Tuesday, followed from a tube that clouded when it should not have. The tube is long gone. The ledger it was written in is probably gone too. The routine remains. That is where we will leave it tonight. A laboratory in the evening, glass, saline, and a lamp. Sleep well. We will speak again.